Now, in this video, um, I'm going to be discussing kind of almost like a hybrid approach because, to be honest, APE is my secondary kind of tool I like to use. I really like Benchling just because the interface is easy to use. I feel like sometimes it's a little choppy in APE, but APE does have its benefits and can do some stuff that Benchling cannot. But so for this guy, we're going to be going over how to analyze your sequencing file. Okay, so the sequences came back, spoiler alert, and we are in business. So we are definitely cooking. So I'm going to be posting links to the data. All right, I'm going to be posting links to the data and stay tuned for that. But what you're going to do is we can import this stuff. All right, um, let's actually go to APE first. Okay, we're gonna go ahead and we're gonna open, and this is also I posted this in the on the website on the Brightspace, so you have an annotated APE file, same thing as Benchling. But okay, let's see. Let's go to open, and you're gonna see a whole bunch of files like this. Okay, actually, let's go back a level. Right, there's two files I'm going to give access to. The seq file, which is just the annotated sequence, and the ab1 file. Actually, I was just going to post the ab1 file because it's redundant, and you can get this information from this file. So you're going to see a whole bunch of ab1 files. I've already made a couple to use as an example. So we're going to do an example. Let's go. We're going to do test 2 first, right? Photo system 2, then photo system 1, right? The order of the discovery. So now we're going to go to test 2, and then we're going to click that thing. We're going to click that thing, and it should pull up a window. Okay, here it is. Test 2, and this is electropherogram, I believe it's called. Like usually with Sanger sequences, the first couple of nucleotides are out the wazooski, right? It's just not a good look. Okay, so you're gonna have a lot of noise in the beginning. That's totally normal. So what we're going to do is we're going to ignore that and we're gonna go and look at our internal file, right? And you're gonna see these humps, and each of these humps represents, depending on the color, a different nucleotide. And on top, we have the nucleotide position. All right, so this is really, really cool. This you can give it a quick gander at to see if your sequencing file looks good. This one looks very, very good. I'm going to do also an example with one that doesn't look so clean, okay, and what you can do there. So if you want to stay in APE, it's very easy. You can go ahead and go to File, and then you can go New DNA from base calls. And here you have a file with the double-stranded DNA and the base calls. And then you can do what I'm getting ready to do in Benchling now, okay? In APE. But I like Benchling, so we're going to go back and forth, right? And I feel like it's helpful for those of you that want to either use APE or Benchling, right? Because you're going to see both at the same time. So what this is, you're doing a little scan. It looks good. Now let's go back to Benchling. Right, and then what you can do too, Benchling also can handle these files, but it's not going to give you the electropherogram, right? So we're going to import DNA RNA sequence, right? And we're going to choose a file, and we're going to choose our test2.ab1. Okay, it's going to load, and this is not going to give us the electropherogram. It's just going to give us the sequence, right? APE can do this as well, right? So fret not. So what I like to do is also what's pretty cool is you can actually highlight this sequence, and then directly from Benchling you can submit to BLAST. So if you're doing a novel kind of sequencing project and you want to make sure you got what you're looking for, you can submit to BLAST. It'll open this BLAST up, nucleotide BLAST, and all you gotta do is click BLAST to see what happens. and it's going to look for the closest match. And hopefully, if we're lucky, hopefully we'll get our gene of interest. But hopefully it doesn't take too long because this is not a good look for the video length. Okay, so it's taking 12 seconds. It's going to update. Maybe if we hit there it goes. Okay, and then what do we got here? The top hit, right? See, 100% identity. There's a couple other ones. But our top one is what? TAS2R38. Would you look at that? So that's a good sanity check, right? So let's go back. Let's go back to our Benchling file, right? What can we do? All right, how can we get to our sequence on the electropherogram from this just nucleotide sequence here? So what we can do is, this is pretty cool. I like this. We're gonna go here. We know what we're trying to look at. Our SNP is in this region, right? It's either gonna be GCT or GTT, or if you're a heterozygote, right, both a G and a T. Right? I mean a C and a T. All right. So here we go. How do we do this? So what I like to do is I like to take a little piece of sequence right next to where I want to look at, and I want to copy that. Right? Click copy. Get all these different options. Pretty cool. Click DNA sequence, so it's copied to my clipboard. Then I'm gonna go to our AB1 file, and then I'm gonna search for that sequence. Control F or the magnifying glass and paste. And there is our sequence, right? There is our sequence. And right next to here, because remember, we right next to our SNP, right? Right now, oh man, I lost it, but we can find it again. Right next to here, right? And then you see GCT. So it's saying this person is a taster, right? A taster, but we got to confirm that, right? Because they could very well be a heterozygote. So how do we do that? So on this file, right, there's a position, the insert position, right? 209, 209. So we're gonna go back to our electropherogram, and we're going to look for position 209, 210. 210, 209. Here it is. GCT. I guess it's right here. Is that right? Yes. And then the nucleotide GCCT. Yep, this is where we're at. GCT. And let's zoom in. So with this thing, you can go here, you can go here, and then we're gonna go back. 209, right? 209. Look at that. 209. And it is indeed GCT, okay? GCT. So this person has the wild type taster copy. How about that? How about that? Right? So then this is how you know, and you can write about this on your report. And actually, there's a spreadsheet where you're going to write your genotype for the sequencing experiment, the genotype for your restriction digest, and whether or not you actually taste the compound. All right, so that's one. Let's do another example. Let's close out of here. This is test 2. All right, this is test 2. You could do the same thing just to be thorough. All right, just to be thorough, we could do the same thing. Right, in APE, right? We'll take a little piece here, copy, and let's see, we have our little window. Guess we can find, where's the little magnifying glass? Find, and then can it find it? Here it is. Find next. Here it is, right? Right at around 209, offset by 1, right? 209. So same business, and you can go on the electropherogram and look it up. All right, so the next one we want to do, we don't want to save that. The next one we want to do is another example. This one's just called test. Let's open this up. All right, opens up. Okay, this is not as good as the last one, right? There's a lot of noise. Same thing. And then we have smaller kind of bass calls. You know, it looks kind of weird. We got this little hump here. We could even zoom in. It's not as clean, right? And they gave me like a detailed report. This one came out as like poor sequence quality. But again, what that means is just that the machine sometimes doesn't make correct calls. But what you can do is if you have a poor sequence quality, you can actually go into the file yourself and look for different nucleotides to make sure it's calling the right bases. And for ambiguity, let's see if we can find one. This is a C, T. This is a G. Yeah, this looks like it's doing it okay, right? But in any position, you can always edit the nucleotide up top to see if you don't believe what it's saying, right? Like here, for example, right? It's calling this thing N, right? But we can see that it might be very well be right in an A, right? It's green. A's are green. Or yeah, A's are green. Okay, so okay, that's one thing about this, right? So okay, we got the sequence file. It may not be look the best, but what do we do? What do we do? Okay, let's see. We can go to the. Let's see if we can do it here in Benchling. I mean in APE, new DNA from base calls, right? And then can we search on NCBI here? Yes, you can. You can also do NCBI. So you know what? We can take a piece of this thing. Let's take a big piece. Right? It has a lot of N, so it might not, work as planned. But okay, no, it's not. All right, here it is. Blast sequence at NCBI. Blast program. Okay. Selection only. Low complexity filter. Expect. All right. We're gonna keep all that just default and see what happens. Let's see what happens. Okay. Oh, yeah, okay. Receptors, partial coding sequence, 95%, 90% identity, right? It's still putting up the right thing. Well, not almost. Yeah, this is a little different, but we're in the ballpark, right? It's not too far off. It's not too far off. Okay, here's our taste2 receptor. So it's on the list. Okay, so again, right? See if we could salvage this file. See if we get our snip. All right, let's see. Okay, this was a pop-up, so I can close this. All right, so then now we can go back to. Okay, let's do it in Benchling. No. Let's keep it in 8. It's gonna be too long. All right, so what we can do is the same kind of thing. Let's see if we can search around. Oh, now my cat is acting up. Okay, let's see if we can copy. Control C. My right click is not working. I don't know if this actually copied. Yeah, APE gives me problems sometimes. And then now, let's see if we can search here. This little magnifying glass. Paste. Okay, find next. Okay, I actually found something. We're in business. Right. So, is it right? G, C, T, G, C, T. Yep, right on the top strand here. G, C, T. And this is on position. Okay, let's find next, close, G, C, T. And this is position 214. 214. So let's go over here, right? 214. Okay, we're in the ballpark. Park. To 10, 11, 12, 13, 14. G, C, T. But let's have a closer look. Right, we're going to boost this up. Right, so this is G. Same G. We've seen a G, right? I'm pretty confident that's a G, right? But here in the C position, huh, could be a C, but it also could be, right, a T, right? They're very close. This may mean, right, that this might be a heterozygote on our hands. The nucleotide right next to it. Look at that. Got called perfectly, right? It's definitely a T. So this here, I would say, right, I would say maybe, just maybe, this is a heterozygote. And what we can do is we can cross-reference our other data, right, to see if this is consistent with this idea, right? So this is just a little tutorial on how to go ahead and look at your sequencing file and make sense of it. But I hope this helps. And for your homework, right, you're going to have to fill out that data sheet, right? You're going to say, you're going to look at your electropharogram, you're going to screenshot this thing, right, in the position where you believe that the SNP is located, and you're gonna make the call. You're gonna say, listen, based on my gel, based on my taste test, and this experiment here, I believe this is what I am at this locus, right? So pretty cool. And we're gonna be doing the same thing for another gene as well in the future. So I hope this helps, and I'll see you soon. I'll see you soon.